So just to quickly remind you that we will be recording the session and it will be available in the YouTube for people who have missed, the session. And some of the ground rules, just feel free to, unmute yourself anytime and ask questions, or, if you have any suggestions, please feel free to interrupt in between. The other thing is, what else am I forgetting? Yeah, in the end, we will probably take a group picture. So again, if you are okay with us taking a picture, please, you can switch on your camera. If not, that's fine too. And today we will go through our sort of the first workshop day, and we're pretty excited, and thank you for joining us and welcome everybody. Just quickly to go through, what stuff we have sent you. We wanted to go through the checklist. Iqbal, if you might, yeah. So this should be the checklist which you got along with the stuff on the right. So has everyone received this? And there should be PH paper also along with this. So that's missing in the checklist over here. Does everyone have that, or is anybody's kit missing anything?
Yes, Sankita, we got everything from the checklist.
Okay, perfect. And everything is fine, nothing broken, because we know like there is some stuff which had to travel a long way. So we were hoping that you have received everything, Okay, perfect.
No, everything was fine.
Sounds good. So we are good to go. And next I will hand it over to Iqbal to start our session today. Go ahead, Iqbal.
Thanks, Ankita. Let's see. All right. So really happy to host this session. And we at Openfield Collective are super excited to dive into this workshop and do this wonderful project with you all. Okay, so today's contents are going to be the following. We'll talk about the kingdom of living things and how living things are classified. Some of the concepts or ideas that we'll talk about are or could be very basic, but given the diversity of this group in terms of age and knowledge base, we thought we would include more or less everything and try to connect the dots and eventually narrow down on why we are doing this project together. We'll talk a little bit about microscopy and basics of microscopic life, and we'll talk about the different kinds of cell types, plants and animals. We'll talk a little bit about photosynthesis, which I'm sure all of us have heard of, and then we'll jump into algae and algal bloom, which is the primary agenda of this meetup. And then we'll explore the algal web that we have created at Openfield Collective. Okay, so the first thing is let's try to identify how living things are segregated on planet Earth, at least as far as we know that the only biological life that is available in this universe is on planet Earth so far. So how is it segregated? So Robert Whittaker, who was an American ecologist, basically described or came up with the idea of dividing living things into 5 major kingdoms. One is animal, which we all belong to, all of us here. And then there are plant, and then fungi, protista, and monera. So it is important to take a note of all these, because the things that we'll talk about today will span at least 3 or 4 kingdoms, because it's so diverse and interesting is that they cannot be assigned to a particular kingdom. So we'll talk about multiple kingdoms. And let's also take a look at the length scale of life, starting from an atom and all the way to, let's say, bigger organisms like human beings. And in that entire spectrum, there is a whole variety of living things in between. And as you can see, the dimensions go from atomistic scale or 0.1 nanometers all the way to meters, right? So there's a huge diversity of life on this planet. One of the most important and abundant life forms are basically microscopic. So they lie in that domain. I hope everyone can see the cursor that I'm moving. So the the unit here is micrometer. So it's a scientific unit that basically is 1,000,000 times less than a meter. Okay, so we'll also talk about what a micrometer is. If you have any questions, you can also ask me what a micrometer is. So let's try to see what a micrometer is. So this is basically a ballpark schematic of how small is a micron. All of us have had coffee at one point in time, right? At some point in time. So coffee beans are basically millimeter scale. You can see them. And then you have sand, table salt, human hair, which is around 50 microns, okay? It's pretty small, so you get an idea of how much 1 micron would be. And then you see pollen grains, which I'm sure we all have seen or have heard of. Then there is limit of visibility. You cannot see things beyond that, smaller than that with your naked eye. And then there's this entire regime of cells and bacteria, viruses, which fall below the limit of visibility. So that basically sums up a large variation of size and how small a micron could be. Okay, we'll talk a little bit about microscopy today. And since we are in the going to talk about the world in the microscopic domain, we need a tool to see that, because we obviously cannot see things with our eyes at that scale. So why would one use a microscope, and how does a microscope work? So as I explained already, that a microscope is necessary to see things that we cannot see with our own eyes. And you all now have a microscope with you. Even I have one in my hands. Thanks to full scope instruments, they were able to sort of democratize use of microscopy among the masses. And how does a microscope work? So a microscope basically uses a bunch of lenses to magnify things that are really small. And the way lenses work is light basically gets refracted as it passes a lens. Now, a glass of water can also sort of act like a lens. And this small experiment we all have done or seen. Every time we dip a straw in a glass of water, we see that it bends, it doesn't look the same. Or you can put your hand behind a glass of water and see how things can get magnified, right? So a microscope basically uses a bunch of lenses to magnify the things on the other side, and you can then see it.
I think we could ask people one question. What is, in our body also, we have 2 beautiful lenses, except mine, I have 4. Can you guess what they are? Anybody? Oh, perfect. Yes, eyes. So we have those lenses which, are so controlled with the muscular muscles in your eye that, it can accommodate a lot of light. So if there's too much light, it can shrink and expand. So it's amazing how nature already managed to figure out to make lenses for you, right? And then later on, we figured out ways to make these artificial lenses. Sorry, Iqbal, to interrupt. Go ahead.
No, yeah.
I
think, yeah, that's a fantastic example of a lens that is flexible. Most of the lenses that we have on our microscopes are not flexible. So that's a very interesting.
Humans have never till now managed to make a lens which is as good as the lens we have.
Right. So the best part about the lenses on our eyes is that they are flexible, and then they can change the focus, right? That's why on new Apple phones, you see they have multiple cameras with different focuses so that you can get depth perception, which we naturally do for, since humans evolved. So what are the different parts of a microscope? So without boring you too much, I'll just quickly explain what a microscope is made up of. A microscope has an objective lens, and we just talked a little bit about lenses for magnification. And then it has an eyepiece on which you put your eyes, and there's a sort of light path between the lens, the objective, and the eyepiece, so that light can travel from the sample through the objective to your eyes via the eyepiece. And then there is a stage. So that's a traditional microscope that we all have seen in our schools and high schools. It's a light microscope, and then there is a knob that will sort of adjust the focus, as in, like, you know, you want to move the objective and the eyepiece relative to one another so that you can, and also relative to the sample so that you can better visualize what you are looking at. Okay, the microscope that you all have on your hands right now also works on the same principle, but it is much more simplified in order to make it frugal and democratize the use of microscopes. So here we also have an eyepiece, and then the eyepiece and the objective are set into the same frame so that, you know, it simplifies. Obviously, this microscope won't have the capabilities of one of the high-end microscopes, but then it serves the purpose. Regarding changing focus, we have a knob there. It's a mechanical knob that is built of paper in the form of a wedge. You can push the wedge and then change the distance between the lens and the sample in order to focus better. And there are some foldscope experts on this session today with us, like Rafiq and Pandya-Rajan, and they can correct me if I'm mistaken. Then there is a stage, as we know that our sample is like larger than what we can see. So once you want to visualize different edges of the sample, you basically move the stage. And I can show it to you briefly here. As you can see that I am able to move the stage like that. I think there's a problem with, yeah, here you can see I can move the stage. Yeah.
Try to remove your background and then do it.
Yeah. All right, moving on, let's come back to the classification of living things again. So one of the major, and if you have any questions, please stop me. Shall I wait for 2 seconds if you have any questions so far about the foldscope that you have? Okay, then I'll move on. So among the different kingdoms of living things, one important distinction is whether the cells that make those organisms are, whether they have a nucleus or not. Now, what exactly is a nucleus? So nucleus is a compartment where we store genetic material inside all of our cells, right? So we, like all the cells that make up the plant kingdom, animal kingdom, and fungi, and also protista, they all have a nucleus which has the genetic material safely stored in a box inside the cell. The only difference of the kingdom of the species in kingdom Monera with rest of all the different kingdoms is that they do not have a nucleus. And we have a name for them. We call them prokaryotes. So let's just try to remember that name for now. So prokaryote essentially means cells or organisms which do not have a well-defined nucleus that stores their genetic material. And you have seen or you have heard of these prokaryotes, and they are the most abundant organisms on this planet. They are bacteria mostly and archaea. So we'll talk a big deal about bacteria and algae today. And then the other side of the story is all these organisms are called eukaryotes because they have a nucleus in them. And every organism that is there in animal kingdom, plant, fungi, and protista, they are eukaryotes. Now, there's another category based on which we divide these organisms into different kingdoms. And that category is whether they are made up of single cells or they're made up of a colony of cells or multicellular. Like we are built of a colony of cells, right? We have different kinds of cells, and there are smaller life forms as well, but they are multicellular, right? So some of the like algae that we'll talk about today are single cells, and they could also be multicellular. Amoeba is a good example, which belongs to the class of single cell, and then there is paramecium. Now, and they all basically belong to, I mean, you won't be able to see them with your naked eye, so they all belong to the class of microorganisms. So what is the difference between a plant cell and an animal cell? So a plant cell, as I said, is also an eukaryote. An animal cell is also an eukaryote, right? But then the fundamental difference between a plant cell and an animal cell is that plant cells can create their own food. They are called autotrophs. And animal cells, or animals in general, they consume plants or other animals for energy. So as you can see in this flow chart, that plants can do something called photosynthesis because they have a special pigment or an organ in those cells, organelle in those cells, and they can create their own food or sugar, right? Let's not like go into too much complicated details as in like different organelles in those cells. I mean, if you're interested, I'll share the link. You can go through it on your own. But then the fundamental difference is that plant cells have something called chlorophyll with which they can do photosynthesis. Now, just to give you a brief idea of what photosynthesis is, it's the process by which a cell basically can generate food or glucose by using sunlight, carbon dioxide, and water. So if you look at a plant, then the plant acquires water from via its roots, and then it takes up carbon dioxide from the atmosphere and uses sunlight or the energy from sunlight to convert it into glucose. And as a byproduct, it sort of gives out oxygen. So it never thought of making oxygen, but it sort of is a byproduct, and it really is helpful. We all know why. So the fundamental thing that is in all these leafy plants or tiny plants or single-celled organisms which have chlorophyll is this chloroplast. So chloroplast is a small organelle which is abundant in those cells, and then that organelle has a pigment called chlorophyll. So we'll talk a big deal or a great deal about chlorophyll and what is chlorophyll and why is chlorophyll so important. So as you can see, this is a cross section of a bunch of plant cells here, and they all have chloroplast, these circular ball-like structures that you see, and they are green because they have chlorophyll, because chlorophyll is green in color. Now that begs the question that
why do we have so many chloroplasts? So,
it's a good question.
Like they're stacked on top of each other, right?
So these cells have evolved to maximize their food storing and food producing capacity. So if I have a factory and I can make, let's say, one fold scope with one machine, then I would like to maximize the number of machines so that I can make many fold scopes, right? So that I can distribute it to all of you. You see? So that's the whole idea. It's all about efficiency, and evolution has driven them to make more of chloroplasts or to be able to make more of chloroplasts. Now, coming to the question, why is chlorophyll green? And this sort of ties into our next session, but I'll leave you with that thought for some time. But if you have an answer right away, you can just unmute yourself and tell and share your thought with everyone. Are people typing?
I don't see anyone type.
Yeah, so let's keep that thought. It's very interesting why chlorophyll is green. It's super. We all know it is.
So green. Is it really green, or is it just we see it green?
That's a philosophical question. It's actually green. So white light, as we see, like I'm sitting in white light, and if we go out in the sun, it's all white light, right? So white light has three primary colors.
Iqbal, Sandhya is saying it reflects green light, and I think Rafiq has a question. He has his hand raised.
Yes, go ahead, Rafiq.
I think you're muted, Rafi.
No, actually, yeah, you can go ahead. That is, I was just going to explain why
Yes.
it is, but I think my connection is very weak. So maybe Iqbal, you can go ahead.
Yeah, no, I mean, someone already said the right answer, which is they reflect green light. So it is important to note for everyone that chlorophyll essentially reflects green light. That means that they absorb all the other lights that is there in the spectrum of lights in white light, right? So let's keep that thought in mind. We'll need it later. Okay, now, so what are algae and what are cyanobacteria? I mean, if you live near a lake, you have heard of probably cyanobacteria, and we all have heard of algae. So what exactly are they? Like, so algae are organisms which belong to three different classes of animal or living kingdom of living things, plants, protista, and monera. So they span a whole different, like they are extremely diverse in the sense that they could be single-celled, they could be multicellular, and they could be eukaryote and prokaryote. So algae, as you can see, you all have probably seen the green scum on top of a pond or a lake, or the entire lake has turned reddish, or leafy things growing on top of a pond. All these things belong to the class of algae. So there is a bit of controversy, as in, like, do we consider algae as a separate kingdom, or like which kingdom does algae belong to? I find it fascinating that algae belongs to all classes and all kingdoms, three kingdoms in particular. And there is still a controversy going on, like among biologists and ecologists, like how shall we describe algae? As you can see, the algae can be kelp, which are leafy seaweeds like things, diatoms, chlorella. Chlorella is the most abundant, and all the folks from India today, it is interesting to know that most of the algal bloom that you see in your ponds is chlorella in the South Asian subcontinent. And multicellular and single cellular, I said already, and algae can be prokaryotes as well, which explains why we call them cyanobacteria or blue-green algae. Now, a fun fact, as I sort of am going there already, is that algae, are they plants or protists? And what is the difference between plants and protists? I urge you all to go back and do a little bit of Google search. Like, well, many of the things in the protista kingdom look like plants, but why don't we call them plants, right? It's a very interesting question. And what exactly means to be a plant and not a protist? And then algae are single-celled or are they multicellular? And whether they are prokaryotic and eukaryotic. Well, I already sort of answered that algae belong to the three kingdoms, Monera, which primarily consists of bacteria. It turns out that some of the bacteria also have chlorophyll, and then they can produce their own food. So we call them blue-green algae. The protista is basically single-celled, migratory or non-migratory organisms, mostly single cell, but there are some exceptions to it. Some of the algae belong to the kingdom of protista as well. The fundamental difference between protista or protists and plants is that plants have a strong vascular network and they have roots. Some of them have flowers as well. Algae or other protists don't have, plant-like protists don't have flowers or don't have roots. That's why we do not push them into the plant kingdom. And then, of course, the big algae or the plant-like seaweed and all, they belong to the kingdom of plant. All right. So a little bit of history of why algae is important and what's the story behind algae. So one of the oldest fossil record indicate that algae appeared on this planet around like 1 billion or 2 billion years ago. And it's a huge time scale. It's hard to wrap your head around the time scale of 1 billion years, right? But they are that old and they are still here with us. And a major breakthrough in evolution happened when an organism decided to consume a photosynthetic bacteria and then live with it and itself become photosynthetic. So that event sort of created a whole cascade of events where there was nothing there to eat them. So they populated the entire planet and oxygenated the planet for us to live and, for the rest of the evolution to happen. So a major breakthrough in evolution is this rapid oxygenation of the entire planet. By rapid, obviously, I mean hundreds of and thousands of years, but it's rapid on an evolutionary time scale. So algae essentially have given us the gift of life and the biodiversity that we know today. And we all probably have seen algae in our ponds. So they look weird and yucky, but they essentially maintain the aquatic biodiversity and balance because they are like at the very bottom of the food chain, right? So they generate sugar or energy, and then rest of the organisms eat them and survive, right? So the transfer of energy from sunlight to all the bigger organisms goes via algae. But there is a catch here. I mean, algae is fantastic, but what happens when you have too much of algae in a pond or in a system? As we know, proverbially, that too much of everything is not good. Anything is not good, right? So if you have too much algae in a system, then what they do is they consume the nutrients in the pond or in that water body, and they make it hard for other species or other organisms to live, like fish. And some of the algae are toxic as well. So they create or produce toxins, which is unhealthy and can cause severe damage to children and also pets as well. And also it could cause damage to adults. So at Open Field Collective, with all that, I want to say that we want to create a global map of freshwater algal species. As you can see, there are like so many variety of different kinds of algae there. And we will use primarily microscopy to identify and map out the different species in freshwater bodies. At least with the cohort that we have, we all will be assigned a water body, or you will assign yourself a water body, and we'll go out into the field, have some fun, and do a quick experiment. We'll talk about the protocols in the upcoming sessions. And then we also would like to monitor the algal bloom with spectroscopy. That's another tool that we have sent you. It looks like a black box like this. It's seemingly sort of very simple and insignificant, but it does a very good job at identifying the bloom. And we'll talk about how we know the bloom. One way of sort of identifying bloom is to measure the chlorophyll concentration. And as you can see, this is a satellite image of the chlorophyll concentration in the oceans around the world. There is not a lot of information or activity going on to measure or identify algal bloom in freshwater bodies. And that's where we think that we can make a difference together. I think the next couple of slides, I'll just hand over to Kanika, and she can take away.
Hello, Iqbal and everybody else. Thank you for giving us a very good introduction about algae, plants, and now I think we can come to what we've all been waiting for. What are algal blooms? So Iqbal, can you go to the next slide, please?
Yeah.
So as Iqbal mentioned, like excess of everything is bad. And as the name suggests, algal bloom is like when you have a whole lot of algae, which you don't want. And I don't know if any of you have experienced this, and maybe you can share, like if you have gone to a lake or any river around you or something, and you've come across this notice that, it is toxic algae, the lake is closed, or don't bring your pets or anything near you. Have any of you experienced that recently, especially in the summers?
Well, Kanika, when I was collecting the samples for my research work, I have accidentally jumped into the water. After coming to the lab, I have noticed that I have got some sort of itching sensation. Then I realized that, yeah, those are the cytotoxins that have been released into the bloom. So yeah, this was my viewer experience with that.
Yeah, that's absolutely dangerous.
Yeah, and we'll be talking about actually safety measures while we're sampling water in our next lecture. But yeah, this is a very important point, and we'll be coming a little bit more to it. But yeah, I mean, I also went to a lake near me to collect samples for this workshop like a month ago. And after 2 weeks of that, I saw that in that lake, it was like toxic algae. Don't come near the lake. So we will talk about safety measures, which is very, very important. And we'll also talk about like the itching of the skin, what kind of toxins may be causing that. So we'll come to that. So, and this is actually a phenomena which is very prevalent in the summers. So, you know, and we'll talk about why it happens a lot in the summers as opposed to other seasons. So Iqbal, can you come to next slide? So yeah, so if you have seen, oh, there's a question from, okay, how do we get rid of toxic algae? Yeah, we're gonna talk a little bit about that towards the end of it. So keep your questions, you know, like, because once we understand what causes this toxic algae, then we can think about what are the ways by which we can get rid of it. So I'll come to it at the very end of it. So, okay, so yeah, this is what an algal bloom looks like. So there is basically a rapid growth of algae or cyanobacteria in water, which results in this like colored scum. So if you have seen like something green floating in a lake or a water body around you, it's like a massive green, which can come in many, many different forms. So these are some pictures from a lake in Ohio and a lake in Florida. And we'll be actually coming to Ohio a little bit, but, you know, due to actually the algal bloom in this particular Lake Erie in Ohio, there was a time when the everybody in the Ohio County were asked for like almost a week that you cannot drink water from your taps. So just imagine the extent of it, you know. You don't have drinking water, you cannot use tap water for anything. So just don't open your tap. That was the thing. And we'll come to a little bit about it, why this happened. Next slide, please. So yeah, so just, you know, like there can be many, many kinds of appearances of these algal blooms. And so these are some of the non-toxic cases. So, you know, if you see anything more on the side of plantish, and we'll be asking you to actually go to a water body near you in the next couple of weeks and just take pictures of how things look like so that we can correlate, you know, whether these might be toxic or not. But if it has more appearance of, you know, like all of these kind of things, like a rooted plant or a plant like algae, like these, you know, if you see anything of a resemblance of these plants, then they're probably non-toxic in that case. But doesn't mean that they're not harmful for other reasons, but they're probably non-toxic. Next slide, please. What is really, really harmful, because they can release toxic compounds, are these cyanobacteria harmful algal blooms, which are also called cyanohabs. And they are toxic because of multiple reasons, but I'll come to a little bit about the kind of toxins which they can release into the water. So if you come across something which is not like a plant, I mean, you cannot see any evidence of a plant, but, you know, like some sort of a green, you know, like a pea soup or a paint which is floating in the water bodies, or some sort of a scum or a mat, and you cannot, you know, say that these are not you know, individual plants that you can just see with your eyes, but just like a floating mat and blob. So those fall more in the category of cyanobacteria harmful algal blooms. And currently, actually, we really don't have a lot of scientific methods to just tell by appearance whether a bloom is toxic or not, or whether it is harmful or not. We really have to go to the lab and do tests and everything. So it may be difficult in the first place, but I think we will encourage all of you to at least take pictures of what a fresh water body looks like around you, like a lake or a river. And then we can troubleshoot together and think about if it may be toxic or not, if it is cyanobacteria, or if it is just some plants. So we will think about it together. But, you know, it's important to take all of these notes, you know, like pictures or the smell or the things like that. So those are some instances by which we can kind of get to an idea whether they're toxic or not, or whether they're algae or cyanobacteria. But eventually we have to do much more rigorous tests in order to establish that. Next slide, Iqbal. So why? So let's talk about these toxic cyanobacteria, which are causing a lot of menace to the society at large today. All right, next slide. So what causes these algal blooms? So as Aparna had mentioned in her chat, this process called eutrophication. So what is eutrophication is when you are filling up, the notions. Oven has a message. There's a lot of algae. That's amazing. I think, Oven, if you can take some pictures next time when you come and share it with everybody, I think that'll be really cool. Are you up for that?
But I think, yeah, without getting too close.
Yeah, without getting too close for now, because then we can identify whether it's toxic or not. And we can find out, like you should wear gloves or something like that if you want to collect samples, or with your parents and everything. So yeah, so Aparna was mentioning about eutrophication, right? So what is eutrophication? Eutrophication is when you're filling up your water bodies with a lot of nutrients. So, like anything, so we need nutrients to survive. Similarly, these toxic cyanobacteria or algae, they also need nutrients. And the source of nutrients from them is nitrogen, phosphorus, all these elements which are found in the atmosphere. And how do these, in the surroundings, so how do these nutrients get into the different water bodies? So initially, going back a little in time, we used to have a lot of wetlands and a lot of forests and everything. And if any runoff is coming, let's say from agriculture, from industries of all of these nutrients, then the forest and everything would be able to take all of these nutrients, so they wouldn't fall into the water bodies. But now what is happening is that we're using a lot of chemical fertilizers. So if, like in terms of agriculture or even in just normal households and everything, and those chemical fertilizers, they contain a lot of nutrients. So they contain nitrogen, phosphorus, and algae thrive on them. They grow a lot if they're supplied with a lot of nutrients. And this is becoming a problem, especially why summers? Because in summers, you're using excess of everything, and for your plants to grow, or for all of these nutrients to survive, and all of these industrial or yes, definitely industrial affluents, as Uzma mentioned in the chat. So all these industrial, agricultural, household, everything which is getting dumped into these water bodies, the fertilizers or any other kind of sewage, they are supplying these algae with all of these nutrients for them to grow. Next slide.
Kanika, I'm guessing also sewage water and stuff also contributes, right? Because a lot of places, they just release that.
Yeah, I mean, to be honest, whatever waste we excrete also has lots of debris from food particles and things like that. So they also supply all these nutrients to cyanobacteria and algae. The other thing which is causing these cyanobacteria to grow crazily is climate change. And I think we are all at a point where we know that all our human activities and everything have really led to a changing atmosphere, changing temperature. So one of the main outcomes of climate change has been the rise in temperatures. And again, cyanobacteria love good temperatures. So in the summers, when the temperatures are high, again, the algae and the cyanobacteria, which live in these lakes and rivers, they start growing more because they like those temperatures. And the other thing is, building dams or these other effects and other human activities. And what happens is that when you're building all of these dams and everything, water comes to a standstill. So, generally, if you have a water body, there's a lot of mixing going around. So, if you have read some of these concepts of physics, so if the temperature, the hot water from the bottom may rise and then cools and then it goes down. So there's a lot of sort of mixing of the water going on in the water bodies. But due to all of the human activities, the water comes to a standstill. So it's not able to move. And this is again very good for cyanobacteria because they like light. So they like to stay on the top. And if there is no mixing or anything happening, those cyanobacteria will stay on the top. They will take up all the sunlight. They like the nutrients, the temperature, and they'll keep on growing. And then everything which is exactly. So Gunashweta also mentioned stagnant water bodies. So yeah, and everything which is at the bottom of the ocean, then it won't have a chance to survive. So these are some of the factors which are causing excessive growth of cyanobacteria. So we should remember the excessive use of fertilizers, rising temperatures due to climate change, and other human activities which are causing stagnation and severe algal bloom. Next slide, Iqbal. So coming to the harmful effects of cyanobacteria. So the thing with, the cyanobacteria is that maybe if there are only a few in the water body, they may not be so harmful. But when these sort of bloom and they're present in a very large quantity, eventually they start dying. And when they start dying, what is happening is that the cell is breaking apart. And once the cell breaks apart, it releases a lot of toxic compounds. So if you can imagine, if you have much more cyanobacteria, and like in cyanobacteria bloom, you'll be releasing much more of these toxic compounds. So here on the slide, you can see, like maybe Iqbal, you can point, you have these, these like microcystin. So this is one of the most popular toxin, which is, produced by cyanobacteria. And this actually causes damage to the livers. If, I was showing you that picture of Lake Ohio, which had all the algal bloom and everything. And the toxin which was responsible for that particular bloom was called microcystin. And this is one of the most common toxins, actually. Yes, the bacteria or the family of cyanobacteria is called microcystis in that case, which release these microcystin. So yeah, so in Lake Ohio, this was the particular cyanobacteria that was blooming, and it was releasing a lot of microcystin into the lake. And then they had to issue an advisory that you cannot use tap water for drinking. And this also happened actually in China first in 2007. And, it has been happening in Lake Ohio since 2014 every year, almost. These kind of rising microcystin levels, especially during the summer. And it's not getting any better. And maybe at the end of the class, we'll send you via email. It's a very nice educational sort of a comic and a resource, which will tell you how Lake Ohio really looks like and what kind of activities people are trying to do to monitor the algal bloom and get rid of them. So Vasavi has a question: what kind of algae could be growing in our water bottles? Yeah, that's a very interesting question. And actually, there can be lots of different kinds of algae, as Iqbal was mentioning in his slides. And at the very end of our lecture, actually, Iqbal is going to show you a resource, which is, we have a web resource of different kinds of algae and their pictures. So it's a lot of them, actually. And you can click on those pictures and get an idea of what kind of algae may be there. Iqbal, can you go to the previous slide, actually? Sorry. So yeah, that is a very, very good question. And we'll talk a little bit about identification of algae. So Sindhura has another question. Microcystis, what does LR represent? Where is LR? Sorry, here. So this is yeah, sorry. I mean, so you don't have to go into a lot of details about this, but this is just sort of if you have read about chirality in chemistry or anything. So these are like just the left and the right configurations of microcystin. But yeah, we don't have to remember a lot for the purpose of this talk. But yeah, these are just the different configurations of the molecules, because as you see, like these are very, very, like structures of just carbon, nitrogen, and everything going together. So if there is, so microcystin by itself can also have a lot of different varieties. So depending on if there are few changes to the structure here and there can be different types of microcystin level rotation. That's correct. So, okay. Let's get to the point. Okay. So just talking a little bit more, and we have another question. What happens if someone ate a fish from one of these lakes? Yeah. So that's a very good question. So the thing is, as you can see in this image here, like in 2016, there was this whole bloom of cyanobacteria, which causes death of 23 million salmons in that particular water body. So so these fish probably died from the toxins. And obviously, if those toxins, maybe if a couple of fish were to survive and, like they were to make actual fish with it or something, and if you were to eat it, probably those toxins are going to go to your body. So depending on the concentration of the toxins, it's very probable that they're going to harm you. So yeah.
Actually, there are some studies where they talk about how, fish over time can get these concentrated toxins in their body. And it's actually much worse because it's like a bag of toxin which is reaching you and you're eating on your plate. So it's pretty important to, notice that where your fish is coming from, and it doesn't come from a region where there are toxic compounds like this, because they can't really digest it, right? So if, as Kanika said, they manage to make it to your plate, they act like this concentrated source of that toxic compound.
Yeah, and as Shivakumari also mentions in the chat that algae, they're the primary consumers, so they do play a role in biomagnification. Yeah, so whatever effects you see in algae, they sort of keep magnifying down the food chain. And, like some of the other toxins are, like these nodularin, and these are just 3 which I mentioned, but there are many, many more toxins. And nodularin also causes damage to the liver. There's another toxin which is called anatoxin, and that has been called a very fast death factor. So they have done experiments with mice. Scientists have done experiments with mice using anatoxin, and what they found that, if you give this particular toxin to mice at a particular dose, they can die within 2 minutes. So they can be very, very dangerous. And, just to mention that recently in US, in Washington, like in Spokane County, there was a case where a family who had 3 dogs, they went to a lake and the dogs went for swimming, and 20 minutes after that, they died, all 3 of them. So this is really, really dangerous, And as Gunashweta mentioned last time, that she had like skin irritation when she went into the water body. And those skin irritations can be actually caused by the outer layer of the cyanobacteria. So remember, cyanobacteria is actually a bacteria, and they have some structures on the outside of their cell. And if those structures come in contact with your skin, then they, or any other part of the body, they can cause irritation as well. So yeah, so these are, like different toxic effects which can be caused by the algae. And Iqbal, next slide. And this is sort of on a broader scale, like those were most of the toxic compounds, but like overall on the ecosystem, the kind of effects they have, because what happens is that if you have all of this bloom near the top of the lake, near the top of the water body, then what will happen is that eventually all those cyanobacteria die, because they can only grow to a certain extent. So they die, they release toxins, which is one effect. But what happens after they die is that all the biomass, all the material, it settles at the bottom of the ocean. And just the decomposition of cyanobacteria actually takes up a lot of oxygen. So you can imagine, even though when cyanobacteria were alive, they were producing a lot of oxygen, but now they're dead. So even to just decompose them, it takes up a lot of oxygen. And this, what happens is that due to this, we create sort of a dead zone. So in the lakes, at the very bottom, then in that case, there is a zone where there is no oxygen, because every oxygen is just all the amount of oxygen is going for the decomposition of cyanobacteria. And this just creates a havoc in the ecosystem there, in the sense that, all the fish or the plants which need oxygen to survive, they cannot in that condition. So the entire food web can be disturbed by cyanobacterial blooms. The other question is, does algae play a vital role in biomagnification in comparison to plants, as we have more algal plume than plants? Yeah, actually, that's a very good question. I don't have a good answer for this, but I assume the answer would be yes, just because
especially in the aquatic system, right? Yeah.
Especially in the aquatic system, because you have much more algae compared to trees or plants on the lands, as we see. So, and as Iqbal was also talking about, the history is that like the first organisms which actually gave the earth oxygen, they were in the water. So the algae, or which are called, if you have read it maybe in your classes, Prochlorococcus, which was like the major cyanobacteria which inhabits the ocean, which produced oxygen 3 to 4 million years ago. So a lot of sort of balancing of the ecosystem happens in the water bodies in the aquatic ecosystem. So that is very important. And I just want to like briefly mention about Owen's question, which was, how can we treat toxic algae bloom? So to be honest, there haven't been a lot of steps which have led to immediate remediation of all of these blooms. So you can imagine, like, they come every summer. And it's not proper to give like analogy of the SARS-CoV-2, which is like, persisting, but the fact that they're so abundant, like if we were to reverse climate change, maybe we can think about that, we may sort of get rid of these algal blooms. But the thing is, they have been around for actually many, many years, like hundreds of years. And they haven't been so toxic in the past. But somehow now we are seeing, due to other maybe changing activities of related to climate change or human activities, they're becoming more and more toxic. So like, some of the steps which the government agencies are adopting, and eventually in the course, actually, we do plan to have some sort of monthly meetings with some of the governmental agencies and people who are actually managing these cyanobacteria. So we will talk more to, more with them as well, Arvind. So it'll be really nice if you could come to those guest lectures as well. But some of the things, like I can tell you that some of the water bodies are trying to adopt is like, so as we mentioned, like if the water is stagnant, then cyanobacteria start to float at the bottom or at the top of the water body, and then there is this whole stagnation of water and there's no circulation. So like some of the steps people have been doing is sort of artificial mixing. So they have these pumps by which they can just like keep pumping the water up and down so that cyanobacteria don't stay at the top and they can go in the bottom, and some other algae, useful algae, something they can go at the top. So there's like some sort of a circulation of the ecosystem going on by these artificial mixing or pumping methods. But the thing is, they are very expensive, as you can imagine, and then you have to keep doing them. You cannot stop that, because once you stop using that pump, then again, the water body will come to stagnation. So this is not like a long-term solution, and they take up a lot of energy. So those are some of the other changes. And the other steps which some of the states have taken is that reducing the use of chemical fertilizers. So I think around 11 states in US, actually, they have banned the use of some chemical fertilizers in households. So which are releasing lots of excess nitrogen and phosphorus in the water body. So the idea is you want to restrict, or have some regulations, like unless you are producing agricultural resources for a large community or something, maybe you won't be able to use so many fertilizers for your particular like industry or household or things like that. So that is other step which the government is taking, like regulation of fertilizers. Ankita has a question.
No, Gunashrita has a question.
Okay, go ahead, Gunashrita.
Nothing. Yeah, just was agreeing with you that either we have to utilize the artificial waste for cleaning the lakes, or you can wait for the nature to do its work. Like we can wait for an entire season, like, oh, let's wait for the rains and the winters that the water just washed out, and we can can stop the mixing of the industrial waste or the household waste into the lakes or rivers, so that we can control the excess growth of the cyanobacteria or all the algal. Like you have already mentioned this point, but I was just listening to you.
Yeah, definitely, that's very important. And the other question from Nikhil is, are swamps and marshes more or less prone to algal bloom? I am actually not sure about that. I mean, it's a very good question, because I think, because depending maybe on the conditions, does anybody have?
Yeah, I think any stagnant water is prone to algal bloom, but there has to be a very a sort of unique environment in terms of temperature, pH, and nutrient condition in that water body to enhance the bloom or sustain the bloom. Okay, so most of the actions that we can take at this point in time is identify whether there is a bloom or not and take preventive measures. So as Kanika mentioned, that we do not necessarily have a remedy or exact solution. How do we do? We can filter the water, and if it is a small water body, but that's a huge and expensive task. So yeah, most of the action right now is going on from a preventive side, and also if we can find out what different kinds of algae are out there. So cyanobacteria is one, and they're very small, not really visible even with a microscope that we have. So most of the algae that we'll be looking at or non-toxic but still dangerous to aquatic ecosystem when there is a bloom are like much larger, in the sense that larger than a bacteria, things that you can observe under a microscope. But they are also difficult. One thing I'm not sure if Kanika mentioned is that even though they do not produce toxins and they cover up the entire surface area of the lake, let's say, then they prevent sunlight from going into the lake, right? Yeah. They prevent oxygen from the atmosphere to mix with the lake and create dissolved oxygen. As you already know, that aquatic organisms do not breathe, they consume dissolved oxygen. So even though they do not produce toxin, let's say, but their presence or their excess of algal bloom or algae is not really healthy for rest of the organisms. So there is a sweet spot at which we have to stay. So yeah.
So also coming to Owen's question in the beginning about, what we can do. So as Kanika mentioned, like, it's not easy for us, or we don't have that sort of resources or training to go and filter water as, but what we can do is, like, if we understand that there is a water body close to us, which is undergoing some sort of algal bloom, we can always either raise awareness around saying that this is happening, or notify the authorities who are responsible for taking care of that, and that's what we want to do, because a lot of this goes sort of unnoticed, or we don't realize that it's our actions which is causing this. And even if we achieve that, that's a very, very big thing.
I know. And I think like the whole idea is that, because I'll show you, maybe Iqbal, can you go to the next slide? Yeah, so this, as you can see, because most of us are either in US or in India. So whatever green dots that you see on the US map, so those are all the freshwater bodies or, other sources of water bodies where algal bloom has been detected. So as you can see here, it's a lot, especially if you start going near the coast, like in Boston, Maine area, like there are so many of these, like they're just concentrated. Florida, those are other hotspots. So like algal, like just imagine the extent to which all of these water bodies have algal bloom at the current moment. And it's and then on the other side, you have this map of India, which is very, surprising in the sense that actually there's not a lot of effort going on to detect algal bloom inland sources. So in the freshwater bodies, which may be found a little bit inland, you can see all these dots which are present near the coast, but we don't know what is really happening inland. So the whole idea with this course is that, if we can start documenting more about our freshwater bodies, like rivers and lakes around us or ponds around us, and the whole goal is that over the course of, the next 1 year, we'll try to bring as many guest speakers as we can from, like who are doing research on algae, or who are doing government, from the government side, who are trying to treat all of these sources. And we can show our results, which we'll do in the next few months, the experiments which Iqbal will talk about more, like how can we measure how much algae are actually present in these water bodies, or what are the different kinds of algae which are present. So if all of us get together and we start documenting all of these sources and compiling them together, we can, just show it to different agencies within our areas and see how we can help them understand this better from a more biological point of view, and how they can help us, or the other environment to sort of really, if we can treat them or, prevent future outbreaks in the summers. So
Kanika, there's one more question from Shiva Kumari. Is there any criteria for dominance of harmful cyanobacteria over the natural algal pool present in the water body?
Yeah, so again, this is a really good question. Yeah. And there isn't, to be honest, any criteria. Again, it depends a lot on the environmental conditions and what are, like in terms of either the temperatures or the kind of water bodies or just the aquatic ecosystem. But the thing is, like, as the environment keeps on changing, eventually cyanobacteria, they sort of dominate. I mean, we can go a little bit into the biology of cyanobacteria. Like, why it happens is that, so these cyanobacteria, they actually have things called gas vacuoles inside them. And due to the presence of these gas vacuoles, so you can imagine if you have like a gas vacuole or something, you are more buoyant. So you can just go up, on the surface of the water body. So many of these cyanobacteria actually have these gas vacuoles, because of which they can go up on the cyanobacteria or up on the water surface. So eventually, if you had to compare a cyanobacteria and a normal algae plant, cyanobacteria, if you provided the right conditions, it'll come up and it'll take up all the nutrients and all the oxygen, and it likes sunlight. So it's like a positive feedback loop, because once it comes up, the conditions are thriving. So it'll keep on, making more of itself and producing toxic compounds. So yeah, that is one thing. Yeah. So I think, Iqbal, maybe you can go to the next slide. Actually, if there are more questions, maybe we can take over before we sort of, yeah. Any more questions?
Yeah. I'm Shiva Kumari. Like, I have if it is dominating, if the harmful bacteria is dominating over the could it be because of the only because of external factors, or sometimes we understood like in the natural phenomena, like we have succession happening, so it could be a part of succession also. When the you understand, I mean, we have a transformation of water bodies into lands, that we call it as succession as a part of natural phenomena. Can it when that process is occurring, will the harmful bacteria, harmful cyanobacteria dominate? Just
Yeah.
now we were talking about the artificial conditions, fertilizers, that is still there, but naturally, can it occur? My question.
Yes, of course, I can take that question. Yeah, so it can occur, and it depends on many factors. Like everything in biology, I mean, it's super complex. It's not a linear path from one A to B. One important factor we should understand is that when we talk about cyanobacteria, they are algae, but they are bacteria at the end of the day. So their biology helps them to amplify really fast. Okay, so the bacteria divide really fast and they can grow and multiply as compared to other organisms like single-celled algae or multicellular plant-like algae, because they take time to grow and make more copies of themselves. So if there is a advantage to bacteria, then the bloom happens rapidly. And the reason, I mean, now the follow-up question that you might ask, that if bacteria divides really fast, then why doesn't bacteria cover up or take up the whole planet, right? Because there are always negative factors that stop the division. Like my hand right now probably has a lot of bacteria, but I'll just go and wash my hand so it doesn't take up my whole body. You see, so we always have all these positive and negative loops going on side by side, which causes things to go up and down. Temperature, time of the year, all these things are important factors. And also consumption, because there is always a hierarchy of consumption in biology or among living things. So if the predator number goes up, then obviously, the prey number goes down, and there is always a balance. So one of the major concerns that we want to raise or aware the audience here is that human factors can essentially cause imbalance in those in that setting, and that's where most of the problems appear. Otherwise, in a natural setting, let's say, there is always a bloom, and then the bloom gets controlled over time due to weather or other conditions. And does that answer your question, Shiv Kumari?
Yes. And one more, that is it the limitation of the bloom is just because the oxygen levels goes down? Maybe because of that. Though
Possible.
it's an oxygenic, yeah, because it will be covering. I feel like it will be covering the whole surface. So usually the water becomes less in the oxygen level, and so it stops growing. Is it that?
Yes.
It
Yes.
could be a factor, or it is the only factor.
It could be one of the factor. Temperature is another factor, right? As temperature drops from summer to fall or winter, the viability of bacteria or other things which were thriving at that temperature will also drop, right? So there could be so many factors. So we can all, these are very interesting questions, and we'll do a bit of homework, and maybe we'll talk about some of the topics, some of these ideas, but it's very good that we all are coming together and talking about it, right? Some of us may know more about why bloom happens. But this slide essentially is a perfect segue for what I'm going to talk next. As you have seen that there is not a lot of data about freshwater algal species and freshwater algal bloom in India and other parts of the subcontinent. And even though there's a bit of data in the US, there's a rate limiting step in the sense that it is not easy to monitor bloom or find out what is going on in those water bodies, even though government organizations are trying. But then the rate limiting step is how often you do the experiment, or how often you go there, or the rate at which you acquire data, right? So obviously you cannot have that many people employed who go and do these experiments or find out or even take a picture, right? So these are dots which sort of indicate where the bloom has happened, but doesn't mean that there is continuous data coming out of that. And as you know, with anything that is in the scientific domain, data observation is very important. So we at Open Field Collective want to monitor or measure the algal concentration and the kind of species in different water bodies. With the current cohort, we have sort of like spanned around 12-hour time zones, difference of time zones, right? So we think it's a global.
Sorry to stop you, but I was wondering, in the map you had for US, is it complete, or it still needs those points, right? Like, I'm guessing this is not a complete map of.
No. It is definitely by any measure, it's not complete. So
that's
these are like
one of the reason we need to do it as well, right?
Yeah. So one of the ideas that all of us at Open Field Collective came up with is that let's create a global project where we engage citizens to monitor their environment, because that's the only way to go forward. We cannot have full professional people employed by the government or private sector to do that for us. So if we want to save the planet or do something for our environment, then we all have to spend some time looking at our surrounding, and simply out of curiosity, there is no pressure on you to do any experiment. Simply out of curiosity, just take a sample and see what is going on, and that curiosity will go a long way in terms of not only making yourself and your friends aware of what is going on, but also contributing to this project where we can essentially find out what kind of algae is there and if there is a bloom or not, right? Because it's a long-term project. So we assume that everyone on this session or in this project will participate by finding a freshwater body around them and go and do an experiment, let's say once a month. So with that data, we can create a global map of what is going on in different water bodies across the globe. And we can add to the growing repository of knowledge at OFC, and it will be always open source and free for anyone to get access to analyze the data. You can imagine that having data like that is not only useful for scientific purposes, but also for making policy decisions, right? If I am the governor of Maryland, let's say the state I am in right now, I would like to know where and which are the water bodies which are dangerous and where I should spend much more of my time. But I'm limited by the resources that I have. So at Open Field Collective, we think that using frugal tools and with citizen science, we can sort of achieve that goal. And in that direction, we also have a learning resource called the Algal Web. Let me quickly jump to that. And what we can do, I think we are almost about time and I won't take more of your time. What we can do today is let's try to learn at least the name of one species of algae, okay? So I'm gonna go to Algal Index and sort of, the algae here, the freshwater algae here are segregated with as like with the alphabet of the starting letter of the alphabet, right, of the name. So you tell me what you want me to click on. Anyone?
Z or Z, whatever I call it. We call it a Z or Z.
Oh, Z. Okay. So let's go to Z. All right. Anyone else wants to volunteer on their favorite name? So we have Zygnema and Zygnema 20 micron, which I think is a magnified version. And there's a bit of description here about the chloroplast and shape and everything. I think we have only 4 names in Z. Interesting. Z, Eric.
Somebody already picked Zygnema.
Zygnema? Okay, let's click on that.
Yeah, Dr. Snehalata picked it.
Okay, so this is how Zygnema looks like.
Great.
It's funny. It looks like a miniature bamboo, right? So I'm from the state of Assam, so I see a lot of bamboo there. So so as you can see, this creature is made up of cells lined up in a linear fashion, like one cell after the other, like a train, or and they all have green chloroplast inside them. And that is how they produce energy.
Is this dividing chloroplast or what is it like?
So it looks to me like a cluster of chloroplasts.
Yeah, I don't think it's dividing. I think it's just how it's arranged.
Yeah,
It's
it's
so
just
beautiful.
arranged like a dumbbell.
I have a question.
2 nucleus are there in that cell.
Oh, wow. Okay.
At my collection of these species, I observe these Zygnema.
Uh-huh.
Wonderful. So you have already collected Zygnema previously.
Yeah. Just in that antimicrobial activity, at the time I was collected these species, but I didn't done that activity. Just I observed.
They have 2 nucleus. Nice.
Yeah, that's super
Shiva
interesting.
Kumari also has a question. What are those shining granules?
Here on this image, Shiva?
Yeah, Iqbal. Yeah.
So
I guess those are just the focus things. Like,
I think they're still
the shiny parts haven't been focused.
Yeah.
So only the focused parts have been showing in the green color.
Yeah, what is interesting is that there are 2 nuclei here. So this is one, that is the other one. And they have some sort of connection in between them, right? And then all the chlorophyll packed into chloroplast is like these circular dots that you see are all chloroplast. So they are like clustered around the nuclei and all over the place. So they are not uniformly distributed. I mean, there is so much to know and so interesting cell biology going on at that scale, right? Because the animal cells that we work with, or Ankita, you may have a better comment there, they don't look like that, right?
No.
So yeah, I mean, it's also a learning experience for us. Sure, I mean, if
also how like the symmetry is that, as you mentioned, like they're in a form, but also these are like compartments separate and then stacked together, forming this like long filamentous structure. It's beautiful. Yeah.
Well, in my mind, if I were to design a cell, I would distribute all the chloroplast uniformly so that I have maximum surface area. But it seems like this.
Yeah, but for example, if it has something to do with, like in terms of getting information or passing information to nucleus, that makes sense that it is very close to it.
Yeah. And also it's possible that maybe, the pathway to create energy or food and transfer that energy to do some interesting processes, it's better to stay closer to the nucleus here.
I'm going to write that as a question and we'll read about it.
Yeah. Let's all go back and do some homework. Okay, so we learned about Zygnema. And what is Zygnema? This is it's a filamentous chlorophyte. It belongs to the class of chlorophyte. It's a subcategory of algae. It is unbranched, cylindrical, by the way, cylindrical cells with 2 stellate chloroplasts.
Iqbal, Ankit had a question, and then Sayak also. Yeah.
Actually, Sayak wrote it. I had the same question. So, I mean, excuse my ignorance. You did mention the scale that we can see with Foldscope and the magnification. So, I mean, Sayak asked the same question. So, will we be able to see something like that with a Foldscope, like the 4th image?
Yes. So I will, in the next session, I will show you some of the, some of the pictures that I took of an algae named Chlorella vulgaris. So Chlorella is one of the most abundant algae, and people who are interested in space and everything. So every time there was a space mission or a flight to International Space Station, they sent Chlorella samples because they grow really well, and they are planning to use Chlorella to convert the CO2 produced by astronauts back into oxygen. So we'll talk about that in the next session, but let's take a look at Chlorella quickly. So here is Chlorella. I think there is only one picture here. So there you see. So Chlorella is on the order of 1 micron to 5 micron. You see the scale bar there? Shall I zoom in? Okay. Can everyone see this? Yeah. So they are this circular single-celled algae that are most abundant, and they have extremely high efficiency in converting CO2 back to oxygen. So as you can see, it's very tiny, but compared to Chlorella, and they also don't form filamentous linkages, right? So that they don't look really big. So you have to concentrate them, and that's the image that I'll show you next time. But if you compare that with this image, as you can see, this image is 20 microns here. So even the entire structure is larger than 20 microns. So if you have somehow manage to collect this algae, then you definitely can see it under the foldscope.
Now, maybe we can give them a homework. I think next time, just take a picture of the water body near you from where you're going to sample. So just, if you have some time, just take a picture, and we will share the recording with you, so you can go back and see what maybe you have algae or you have cyanobacteria. Like, just try to identify, and we can talk more in the next session. No, then in that case, yeah.
Yes. So at least we learned two species. One is Zygnema, and the other is Chlorella vulgaris. Okay, fantastic, everyone. With that, let me see if I have anything more. Next steps, as I said, we'll talk about safety measures while going into the field and sampling water. We'll talk about classification with foldscope, how to use foldscope, which is the microscope that you have in your hands. And we'll talk about spectroscopy and how can you use spectroscopy to measure bloom once you get the samples, and we'll talk about the protocols and discussions. But meanwhile, please go and check the algal web on our website and find out your favorite algae. And next time when we talk, all of you should at least say one algae, name of one algae that you think is your favorite. Not all of them look good, by the way, they look weird. so yeah.
Weird is good.
Well, Iqbal, I have a question.
Yes.
Right, in our water bodies, like there is not like a particular cyanobacteria will be present there. It's like will be number of bunch of organisms living together. So when we take off these samples, when we look under the foldscope or the microscopes we are provided with, it's impossible to have a picture of a single cyanobacteria. So it takes me how we are supposed to, isolate them, separate them, and then take a picture and record them, to present to you. So how we are supposed to do that? Because isolation takes a very huge
yes.
time for that. I'm very much aware with that and experienced with that. It almost took 6 months for me to isolate all separate cells. So how we are supposed to do that? How we are going to plan that?
So one way of, that's a great question, Gunashrita. I mean, one way we have thought of that as a problem, like once you go into the field and collect, let's say, a bottle of water, right? So that will have all sorts of things, like things that are on the order of millimeters to all the way to micron scale. So in order to navigate that problem, we have provided you with the right tools. So as you see in one of the foldscope kits, you see you will get this box. Let me quickly change my background.
Also, so I think just, the question is like isolation. So we're not planning to do like lab isolation techniques or anything like that. But yeah, Iqbal is going to talk about like filtration. So we won't go to the lab or just
right. So also to add on, I mean, sorry, Iqbal, I'm interrupting.
No, go ahead.
Like we don't think that we need to go to that level that you have to isolate and be like, okay, this specific one. And I mean, the main thing we are trying to do here is also to sort of gain curiosity. So for sure, if you have like paramecium and stuff like that, which is always there in water, it's wonderful. Take the pictures of those things as well, because, this is a part of the ecosystem and you don't have to really bias so much. Yeah. Sorry, go ahead.
No, yeah. What I'm saying is that one way of segregating what you get in your sample is by filtering. So with the foldscope kit, you have these filters, right? You have these filters of different sizes. We have also provided conical filters that has a mesh size of 100 microns. And then there are 3 more nylon sheets that come with your foldscope. They have a gradient of like 5 micron, 25 micron, and 100 micron. Okay, so we'll talk about all those, how you can use these nylon sheets and how you can use the filter, the paper filter that we have provided. So filtration is going to be super important so that you can segregate things and you don't have a cluster of things in your field of view inside the foldscope, right? That's one way. And the other way is, as Ankita mentioned, other thing that is, it's extremely important to present whatever you see. So the curiosity is at the top of our priority. So all of us are going to present whatever we see. There is a decent chance that we might end up discovering something really cool and new. Okay, so so one of the speakers that will come up later during our session, he is a diatom expert, which is one of the kind of algae, and he has recently discovered a new species of diatom somewhere in southern India. So you see, there's a lot of scope in terms of curiosity-driven discovery, but we do not need the data to be really, really neat in the sense that you segregate bacteria from chlorella and show chlorella versus bacteria, right? Because we might end up writing a paper, but we do not need it to be a super scientific project. It's all curiosity-driven, but I want to assure you that the data that you get is not also trivial. We can make a lot of decisions based on the data. So that's the whole idea.
And also just to stress, I mean, it's one of the whole project is also because we are trying to cater it towards students and everything. So the idea, as Iqbal and Ankita mentioned, is curiosity, and it will be really, I mean, I don't know, like just from the point of view of future, like maybe we're not isolating them now, but maybe some of us will go into the field and isolate in a lab 2 years, 5 years, 10 years down the line. So those are the kind, like that's like the broader vision. But I think for now, we want to understand what is really happening in our ecosystem. And stage 2 would be, once we talk to other people, whatever we have found out about our discoveries, the relevant bodies and the agencies, then we can go to that next stage. But yeah, so that's just to say, I mean, we want to be scientific and we want to be accurate, but maybe we don't want to go to that stage of isolation yet, just because of the limitations of everywhere we are and all the different resources we have access to. But the idea is if we can sort of, inspire some of us to actually go and do that as well. So yeah.
Shall we quickly take a picture?
Yeah, I really encourage everyone to turn on their
whoever
cameras.
is okay with it.
Yeah, if you are okay, we'll take a quick picture. I promise we are not going to misuse the picture. We'll basically use it for
on Twitter.
Open Field Collective Twitter page to generate more interest. I'm
going to pose with my full scope.
Oh, I have a virtual background. Actually, I can do that.
Yeah, I need to remove that too. Is Ankit taking a picture?
I am. So I'll take 2 pictures. So I'll start counting. So 3,
algae.
2, 1. Smile, guys. Okay, and one more. Just one more, because we have too many participants and I cannot fit everyone on my screen. Thank you so much,
Wonderful.
guys.
Yeah.
Thank you so much.
Thank you so much. So just
thank you, Iqbal and Kanika.
Oh, thank you, everyone.
I learned so much today.
And if you have any questions, if not, then we'll meet again next week, same time. And just try to explore the kit that you have, and we'll make you open it up and do experiments with it next week. And if you get a chance, go out into the field, take a couple of pictures. If possible, maybe get a sample, maybe a flower, leaf, a pollen. Rafiq has a lot of pollen grains, I know. So Rafiq can give us some demo.
But I would suggest
don't touch the water or anything.
Exactly.
Like that
I was coming to that. Yeah, whoever is not trained to do that, I know some people have already, been to field and know how to do it, but others, please, really don't touch the water yet.
We'll discuss safety measures and stuff next time.
Yeah. Just take a picture on your phone or something from far away.
Yeah, especially Nikhil and Owen, let's wait for all the safety protocols to happen, and then we'll jump into doing experiments.
So also in the meantime, keep an eye on your emails. We will send, some of the resources which Kanika mentioned. And also we will already send maybe a YouTube video of how your Foldscope works, because we will be covering that in the next one. I know also I will send Rafiq's video. Rafiq has a wonderful video, which I saw along with the other Foldscope videos, which are there on YouTube. So yeah, go ahead and explore all of that to know more about your Foldscope, and we will cover that in the next session as well. Thank you so much.
Bye.
Have a great day. Bye.
Bye-bye.
Bye-bye, everyone.
Bye.
can stop
Thank you.
recording, right?
Yeah. Ankit, go ahead, stop.